thank you all so much for coming, and sorry about all that, but it's been a really interesting and great morning. We had um, a clinical conference with Reprieve and with Priscilla and Peter Hunt and Colette. And then we had, which is really great, Mohammed come and speak to us about his research, and we missed him from the Wistar Institute. And then we had all the CFAR scholars. And I want to thank the CFAR scholars who are over in the other room right now on Zoom for their not only summer research projects with us, this is the CDEPI program, which is great through the CFAR, but it is really to serve historically either Black or Hispanic institutions. So our Hispanic institution in the city is San Francisco State University. So that's how the CDEPI works. And really want to commend John Ceseda and Karina Marquez, who lead that program. So, so this is just such a cool and exciting day because now we have one more thing to happen. And we're going to have a lecture right now, and then actually people are going to be individually meeting with Dr. Peter Kim. And I don't think anyone who does NIH research at NIAID needs an introduction to Peter Kim, but Peter Kim is essentially, Dr. Peter Kim is the head of the therapeutics research branch at the Division of AIDS. And actually, I just learned this morning his kind of more of his history of how long he's been at AIDS, because he started out doing his undergraduate at Johns Hopkins University of Maryland for medical school, and then did his internal medicine residency and infectious disease fellowship at Georgetown. And after that, this long experience with the NIH is he went straight to the Division of AIDS, has done clinical work in and out of that, and is now in charge of the entire therapeutics research program for AIDS. That means not just actually HIV, but really has a large portfolio of grants that he manages with tuberculosis, and then emerging infectious diseases. So actually COVID and Mpox were big aspects of the portfolio that he managed. We all have a lot of interaction with him, really involved in the clinical trials networks, and we're really excited to hear his talk today, which is actually about, we were at NIAID, I was at NIAID the other day, and I said, what are you going to talk on? And he said, there's so much to talk about, but it's more this kind of bi-directional lessons from what we did learn from COVID in terms of HIV research and what we learned also in terms of HIV setbacks. And it's going to be a discussion, so we're starting a little bit early so that he can give this talk, and then we'll have a discussion and questions. So thank you so much, Peter, for coming to be with us today. We really appreciate it.
Thank you, Monica. And thank you for everyone for welcoming me here. I didn't realize this is kind of like the inaugural lecture, I guess, for this building in this room. So I'm doubly honored. And I'm also a little bit embarrassed to say that as much as work as we do with UCSF, this is actually my first time visiting with you all, but I hope this will be the first of many. So, I debated a long time to think about what I would talk about when I came here, because there's so much, there's so much great work that you all do. And of course, we're involved with that. We could talk for days, but I've been personally thinking a lot about our recent experience in COVID-19 and Mpox. And there's a lot to talk about. There's a lot that's already been published from higher levels of government about lessons learned. But for me, I've been thinking more about the personal things that I've learned, but more importantly, how we might take those lessons, because these were hard-learned lessons, and how we might apply them to the things that we're working on now, that we're kind of getting back to our business as usual in HIV, TB, hepatitis, and everything else we do. Because I do think that there are really some important lessons that we've learned, and it would be really a shame if we didn't take advantage of those things. So these are my personal reflections that I'm going to present to you today, just in terms of the lessons learned. I purposely did not talk about how I think we should apply them to HIV, because I'm really hoping that that could be a bi-directional discussion between us. Because I think, I'm interested to hear from what you have learned, what your experience was like, and what you feel are the next steps, how we should apply that in HIV, TB, and so forth, because there's just so much. So with that said, I have no conflicts of interest to disclose. And I just want to be clear that, especially this time, the views reflected in my presentation do not necessarily reflect the views of the US NIH. These are just things that I've been thinking about, pondering, It's not like I've shared this presentation with anyone else before. So thankfully, Monica already introduced me. I'm thankful for that very kind introduction. I'm the director of one of the 4 therapeutic research programs in the Division of AIDS, and as she said, we oversee things to do with HIV diagnostics, therapeutics, and also TB as it relates, especially as it relates to persons infected with HIV. And of course, we are now have an emerging program in the hepatitis space. If anyone wants to talk to me about hepatitis, I'd be happy to do that as well. And most of the time, and before the pandemic hit, as I said, we were focused on people with HIV and those associated comorbidities and co-infections. But when the pandemic hit, we were asked and honored to take on a series of clinical trials for the development of new treatments first for COVID-19 and then for Mpox. And we actually ended up doing 5 different trials, but I'm just going to focus my talk today on 3 of those core trials that we did. The first trial that we did was called ACTIV-2, and it was initiated in August of 2020, so fairly early on in the pandemic. And this was actually a fairly large, complicated phase 2 to phase 3 adaptive platform design trial. And the idea was that we anticipated, there were no treatments available back then, as you know, but we anticipated that there would be a number of new monoclonal antibodies and eventually small molecules that would be coming down the pike. And we wanted to design one trial to rule them all, essentially. We thought it would be better, faster, easier to have one single trial take care of all those drugs in an adaptive platform design, rather than establishing multiple trials as new treatments came along. And so we were asked to do this by the NIH leadership, and we undertook this effort in collaboration, of course, with the ACTG. And because of just the size and the enormity of what we were trying to do, we also brought on a CRO through one of our DATES contracts to help with that effort. This trial recently finished. So it started in August 2020, ended in August of 2023, and ended up randomizing a little over 4,000 participants. The second trial, the PI who's sitting right in front of me, so I'm a little bit shy about talking about it, is the Scorpio HR trial, also known as ACTIV-2D. And we took the lessons that we learned from ACTIV-2 and we applied it to ACTIV-2D. Namely, we decided to slim down the trial size. We made it about 1 single agent as opposed to 8 different agents. We made it just a phase 3 as opposed to a phase 2 to 3 adaptive trial. We actually allowed the Shionogi company who was providing the drug to also take on the sponsorship responsibility for this trial, while DATES maintained the funding for the trial. We continue to work with the ACTG, and again, just because of the size and enormity of the trial, we engaged with the CRO, but this time through the ACTG and not directly through DATES. This trial is still ongoing. There's a little over 1,300 participants enrolled to date, and we're trying to reach a total sample size of 2,000, hopefully by the end of this calendar year. The last trial I'll talk about is not COVID-19 at all, but is the STOM trial. So just when we were in the thinking that, oh, we're done with pandemics for a while, it's just about the lessons learned, and we were literally thinking about the lessons learned from COVID-19 and wrapping up our activities there, Mpox hit. And the NIH came to us again and said, well, we need you to do one more pandemic response trial, and that's a phase 3 trial for this tecovirimat, which is a drug that was developed for smallpox, but we believe has good activity against Mpox as well. So again, we took the lessons learned from ACTIV-2 and ACTIV-2D, and we developed this phase 3 trial. It's a smaller trial. It only involves the ACTG plus impact with SIGA. We are not utilizing a CRO. We just utilize the standard ACTG and DATES processes. But again, despite the fact, as you'll see, we implemented this trial, but within about 1 month from Dr. Fauci asking me to start this trial, I think we had our first patient in within 6 weeks of that phone call from Dr. Fauci. And yet you'll see there were many lessons that we still needed to learn about pandemic response trials. So the first lesson learned, as I alluded to, is that complexity leads to more complexity. And I think it's so obvious when we say it, but it wasn't obvious, as I said, when we started the ACTIV-2 trial. We thought, there's going to be many drugs coming down the pike. Why deal with the hassle of doing multiple studies? And actually, it wasn't just us. Many people encouraged us to actually undertake this trial design of an adaptive platform trial. The potential benefits being a shared placebo that would reduce sample size because we don't need to enroll a placebo arm for every single agent. That we also thought a registrational trial, as you all probably know, has a lot of administrative and paperwork burden that goes along with it. So we thought if we do one trial, it will reduce the administrative burden of all that paperwork, and we'll be able to do it with less work. And thirdly, we thought it would be easier and faster to add new agents as they came along during the pandemic, rather than having to start a whole new trial every single time. Well, actually, the reality is that none of that really proved to be true. First, we found out that building a large adaptive platform trial was very complex and very time consuming, especially because the size of the trial was beyond what we felt the ACTG and DAYS alone could handle. So as we said, we had to bring on a CRO, and that meant developing new processes, new SOPs, a whole new infrastructure for the trial, and that took a very long time. Any ideas about reducing the amount of work? I can tell you that I've never worked so hard to get a trial off the ground in my life. I mean, these were days when, there was nothing else going on, honestly. So it was good, but we were all working 16-hour days to try to get this trial off the ground as quickly as possible. We also realized that simultaneous implementation of multiple arms really put a lot of stress on our sites. And what to us was, simply on paper, it looks great. Just start 4 arms at once. Sites came back to us and said, it's not that simple. And there's a lot of work involved in starting even 1 arm, and never mind 4 arms. And we also realized that with the rapidly changing nature of the pandemic, and as you know, things were changing fast back then. They're still changing very fast, quickly. We needed a trial design that could adapt very quickly. What we needed really was a speedboat, but what we had built was really more of a cruise ship. And therefore, it really made adapting to the changing aspects of the pandemic and requirements of the FDA and other registrational authorities very, very difficult. So again, the lesson was that while an adaptive platform design in principle has the potential to speed evaluation of multiple agents, what we really found that there were many unanticipated complexity that made this really difficult. And just to kind of put some data and evidence to my premise here, if you take a look at, in blue is the ACTIV-2 trial, and in, I guess, orange is the ACTIV-2D trial. Even when you just look at the site level, you can see that the average enrollment per site for ACTIV-2 was 0.9 patients per site per month, whereas it was where it is now 1.32 patients per site per month. And we actually expect that number to go up because we're still in the process of adding more sites. The second thing is that site activation was 12 sites per month for ACTIV-2, and the site activation rate for ACTIV-2D is 13 sites per month. Now you may say, well, Peter, those numbers are actually not all that different. Why do you think it makes a difference? And not only that, but you might think, well, having a shared placebo arm might negate some of these differences. But I have to tell you, number one, ACTIV-2 was implemented during a time when there were no treatments, and the enrollment was for all comers. Not only that, but we actually had the regulatory benefit of this being the #1 trial, priority trial for the FDA and for other regulatory authorities around the world. So basically, we were able to get approval for ACTIV-2 within days, not months. And we were able, and this was the only, hopefully this was the only trial that was really going on, to enroll participants. And again, it was for all comers. Whereas ACTIV-2D, by the time we rolled that out, the regulatory situation had drastically changed. We were back to normal. We were forced to only enroll standard risk participants in the US, even though OUS were enrolling all comers. And sites actually now had a number of competing trials that they had to think about and activate, not just our trial. But despite that, the active2D, in my opinion, and by these numbers, is performing better. And I think that speaks to, again, the difference between a cruise ship and a speedboat and what you can do, and what you're trying to get out of that trial. The second thing is that really what we learned is that to get ahead of the pandemic, you really need to be ahead of the pandemic. And this is kind of like a Yogi Berra saying, I did it on purpose because it's still so obvious when we say it, but back then, again, it was not so obvious. If you think about how we actually do clinical trials and how we implement clinical trials in normal times, what we do is we look for places that have a high incidence of the disease that we're trying to study, and then we go and build sites there and we activate sites there. But what we learned with the pandemic was that that actually cannot work and will not work because the pandemic is moving so quickly. And so basically attempting to activate sites after detection of rising incidence in a certain location basically did not work for either COVID-19 and as I'll show you for Mpox as well. We try to use predictive modeling to basically help us and try to get ahead of the wave, but that also did not work because the length of time that the predictive model was efficacious for was still too short for us to basically build a trial or activate a site in that location. And for COVID-19, high incidence rates also seem to race through urban areas much more quickly than rural areas. And even though I don't have the data here to show that. But guess where most of our clinical trial sites are in this country? They're in urban areas, not in rural areas. So by the time you end up actually developing the trial and activating the site, you basically miss that wave of incidence. And this is just to illustrate that. If you look here, the top chart is actually our enrollment into the active2 trial from the beginning of the trial in early 2020 through end of 2021. And then below the bottom chart is just the incidence rates in the US. And you can see basically that incidence and enrollment into trials follows incidence and they go hand in hand. And so really what you need to understand though is that waves of high incidence generally lasted 3 to 6 months in this country. That's it, 3 to 6 months, things go up and things go down. But if you think about actually the amount of time it takes to to build a trial, but not only build a trial, even if you're just looking at actually activating a site, for anyone that's done a clinical trial, think about how long it takes you to actually get a site activated. It's months and months. Even at pandemic pace, where we were working frantically to get sites activated as quickly as possible, the average rate of site activation, I think for active2D, when we actually had things down pat by then because of our experience in active2, is about 4 months. So you can kind of start to realize that actually by the time you say you hit the go button because you see incidence rates rising, it's already too late because by 4 months later, the incidence rates are going to have come down. Not only that, but early on, we had these discussions of why we still felt that it was okay to wait. And the idea was that even if the incidence rates go down, well, there's millions of people in this country that are infected, that are getting infected every day. Surely you must be able to enroll into the trial. But when you look at the numbers, difference is that really enrollment, again, not only follows the incidence rates, but it's about 10,000 times less than the incidence rate. And so, we were never able to make up the time loss or the enrollment loss from that, having that site activated early on in that incidence wave. Even though we always think, we can do it, the fact of the matter is we were never able to do it. And the issue becomes even more clear when you look at the Mpox. So from the time that I got the call from Dr. Fauci to actually us, again, building the study and first patient in, about 6 weeks time, as you can see. So we got started in August when we decided, these incidence rates don't look like it's coming down anytime soon. We need a trial. We started building the trial. The first patient was enrolled in September. By that time already in the US, and this is global figures, but by the time just 6 weeks later, incidence rates had significantly dropped. And then when you figure in actually the time it takes to actually get a trial approved through international regulatory authorities, our first patient enrolled outside of the US was in Mexico, and that was in May of this past year. And you can see, actually, we basically pretty much missed the pandemic. It's not just us. The world basically missed the pandemic. We weren't the only trial. There were multiple trials that were trying to do the same thing, and we all missed it. The issue now is that it's not like we can give up, though, because we know that the mpox is not going away. There continue to be spikes throughout the time, but having missed that first initial wave, it's now going to take us a much longer time to actually get the trial done and really provide the registrational quality data to the FDA and other regulatory authorities so that we can actually implement TPOXX, with the idea, under the scientific knowledge that actually TPOXX really works, which we don't know at this time. The third lesson is that a diagnosis requires a diagnostic. And again, just it's so obvious when we think about it. But in the early days of the pandemic, you would not believe how hard it was actually to get access to diagnostics to enroll into our clinical trial. There was just no access. All of the diagnostic capacity in this country was being utilized for clinical care. And that makes sense. But in my discussions with those that were in charge, I also have to actually, show them that, yes, that makes sense. But at the same time, if we can't enroll into this trial, we'll never have a treatment. And we're in this cycle. And we can't, unlike clinical care, for a clinical trial, you can't actually enroll someone into a registrational clinical trial unless you actually have a proven diagnosis. And that was something that actually we didn't put enough thought into, in my mind, at the very early days of the clinical trial. And what we saw actually is that as access to diagnostics improved in this country, that actually enrollment also started to improve. And most recently, Dr. Lukemeier can tell you that one of the impediments to enrolling in South Africa and other countries has been, again, the lack of access to diagnostics. And when we were able to provide those diagnostics to sites for diagnosing patients, enrollment numbers went up again. So while we see that the broad availability of diagnostics is critical to enrolling into a clinical trial in general, we also saw that actually linkage to diagnostic programs and how we make that linkage was critical. So early on, we found that other companies had told us that what works is if you run a diagnostic program, and then you refer those participants or those patients, potential participants to clinical trial, it works. So we took that up and we tried to engage with other diagnostic programs nationwide in this country. But the difference between us and them is that at first, we did not actually have an active role of those diagnostic programs for referral, meaning that it was more of a side thing that they did for us. And what we found is that that was not nearly as impactful, as effective at enrolling into our trial, as then when those diagnostic programs had a vested interest, some kind of an active role in the trial and enrolling into trial. And when we had that active connection, enrollment actually significantly increased. Whereas that passive connection was better than nothing, but really we did not see much of an impact. Really, it was no different than just the diagnostics being available to the community. And I think this is really important when we think about diagnostics, because I was talking with Dr. Gandhi about the role of diagnostics and how we think about that in terms of treatment development. And oftentimes it's been my perspective, or what I have found, is that we say it in words, the importance of diagnostics, but we often forget about it in our actual deeds when we're developing new treatments and when we're thinking about rolling them out. Okay, the next is public research requires public support. Again, such an obvious thing to say, but actually during the COVID pandemic, this proved really difficult to come by. During our efforts to actually enroll and get community engagement and the community support for our ActiveTruth trial, we found that actually members of not just the public, but members of our own health community actually oftentimes did not understand the importance of our trial, what we were trying to do, and the importance of them supporting what we did. And in fact, actually, there were many cases where it wasn't just a lack of support, but they actually they opposed us because they thought there were different, better ways of doing it. If you look at the difference during the peak of the first wave in the US, and you just look at the number of deaths they outpaced enrollment by 2 logs. Now you got to remember, it's different than now, right? Back then, I mean, we were hearing on the news every day, thousands of people, upon thousands of people dying daily from COVID-19. No effective treatments out there. Really, the only thing that was out there for patients with COVID-19 was our clinical trial. And frankly, I can say, we had the best treatments out there. And not only in this country, in the world, because we were working with all of the major pharma companies developing new treatments. And yet, actually, when you look at the rate of enrollment that we had in this country, you might expect, a reasonable person might say, well, you would think that enrollment rates might increase, but it did not. You might think that communities that typically, prior to COVID-19, that did not engage in research, you might think they would engage in research because this is the only place where you can get treatment. That did not happen either. What we basically saw was that the same types of people, the same types of communities that engage in research, they engaged with us during COVID-19, and those that did not, did not. I think this just goes to show that we as a research community have a long ways to go to working with our colleagues in the non-research community to educate them, to talk to them about the importance of research, the role of research, and actually building that trust relationship with them so that they understand that we're here to help. We're not here, to try to do anything that's, untoward towards their community. The one last thing that I have to bring up, because Janet Woodcock would be very upset if I didn't bring it up, is that in those early days of ACTIV-2, I mentioned that we were the only trial with access to the main drugs, and that's absolutely true. But at the same time, I think there were like another 1,000 clinical trials that were active in this country and certain parts of the world, testing other quote-unquote treatments. I don't know what all those other treatments were, but what I can tell you is that even amongst the research community, the research response was a very heterogeneous one. And actually, I can tell you for sure that actually that impacted our ability to actually complete our trial and enroll into our study. Lest you think that actually this is something that's someone else's issues and not ours, this is data showing actually the rates of enrollment or non-enrollment among both academic, commercial, and nonprofit research sites around this country. You can see actually that even though mostly we work with academics, you would think the academicians surely must understand the importance of research and the importance of coming together at a time like this. 33.8% of academic institutions who actually had signed up for our trial. So this is not just some survey. These are amongst trial sites that actually said, we want to work with ACTIV-2, we want to enroll into your study. 33.8% of them did not enroll. 43% of commercial sites did not enroll, and 37% of nonprofit sites did not enroll. Again, remember, this is not just like an HIV trial or a TB trial right now when there's plenty of other trials going on, other treatments available. This is during a time when we are the only major trial in this country that is making available novel treatments for COVID-19. This is during a time when thousands of people are dying without treatment in this country, and yet 40% of trial sites that we had engaged with in ACTIV-2 did not enroll a single participant into the ACTIV-2 trial. I mean, what does that tell us? I think we really need to take a step back and understand why. Why was that the case? I think there are many reasons, and I think we need to learn from that. Okay, next is that we realized community engagement requires us to actually step up and engage the community. We can't sit back and actually think that the community is going to come to us and think that that is community engagement. What we experienced was that those trial sites and those efforts where we actually went into the community, and when those sites that went into churches, I mean, we had some sites that actually advertised the site and church bulletin boards, on the weekly bulletin board. Those were the best enrolling sites in our trial. We actually made efforts to actually go into the barbershops and salons to advertise our trial, and those made the difference. The efforts that made the biggest difference was not those efforts that just put the word out there, but in my opinion, those efforts where we actually actively engaged with the patient groups, the participant groups, where we're trying to reach. But more importantly, those efforts where we had the backing of community leaders whose voices, not that we trust, but those whose voices the community trusts. And that made a huge difference. But think about actually what I said before. Many of those community leaders did not trust us and therefore refused to back the trial. I mean, I can't blame them. It's because we have never made a bridge to those community leaders in the past. And now in the setting of this pandemic, we're just reaching out, that is not the right time to do it. I think this really speaks to what we as a research community need to do if we want to see our research accelerated in other fields. Last but not least, I recently read Bill Fogarty's book on his experience in eradicating smallpox. And this really actually just echoed my experience, some of my experience in recent pandemics. And I thought I'd just share it with you. First is that we need to spend the time and attention needed to systematically improve the tools as well as the techniques to deliver them. So we often think about pandemic eradication as more of a public health implementation effort. But what Bill talks about is the importance of some of the innovations that were made during his time in India that actually enabled them to actually eradicate smallpox in India. And this is what we do at NIH, this is what you do here. And I think it's really important for us to think about what are the innovations, additional innovations that we need to actually make the major achievements we have had in HIV and TB to ensure that they are going to have a public health impact. I think I heard that you're talking about the REPRIEVE study. You all have been involved in probably every major HIV study to date, but that's scientific knowledge that we can easily apply to those people that are in the healthcare system, that are engaged with us. But how are we going to turn that knowledge into actual impact for those people that are not engaged in the public health system, that don't want to be seen by us, that don't want to be treated by us? And I think there needs to be additional innovation that's going to be needed to turn that knowledge, our previous successes, into actual impact for those that actually are the most underserved amongst us. The second thing, coalitions are powerful. I mean, I think we would all say this is true without a blink of an eye, without a doubt. But I want to just repeat these two quotes from the book. The secret to eradication of smallpox in India was that the members of the coalition team suppressed their individual egos for the sake of achieving a common goal. And I just, that really struck me because it's one thing to say it, it's one thing to read it, but it's another thing to actually, for all of us, do we really work in an environment where we can say everyone suppresses their individual egos for the sake of achieving the common goal? I don't know. Maybe we do. I think there's more we could probably do towards that effort. The second is that the boundaries between the central government, the states, the districts, WHO, NGOs, public institutions, and private industry were obscured as the team formed with an unwavering focus on the desired outcome. Really, these two quotes go hand in hand. I mean, if we really want to have an impact, if we want our science to have an impact, I think we need to be able to reach across the aisle, reach across different sectors of what we do to make sure that we're working together as a single unit, as a single entity, to make sure that those things that we come up with, that we develop, will actually then have a benefit for the people that we serve. And then last but not least, of course, trust holds teams together. And I do want to end with this because in my mind, actually, this was the one thing that really did come across during my efforts. I mean, working with Annie, working with the ACTG leadership, working with the companies, I can tell you that there's never been a time in my time at the NIH where there was not only trust, but actually we just came together as a group and just got things done. There was no, none of this bickering. There was no, people fighting for position. It was just a matter of this needs to get done. We're going to get it done. And we couldn't have done that if we did not trust each other. And I hope actually that lesson is something that we can continue on as we work towards HIV, TB, hepatitis, and whatever else we're working on. And with that, I'll bring my talk to an end. But as I said, I'm hoping, and I'll take any questions that you have, but I'm hoping we can have a discussion, a bi-directional discussion of what are the things that we can learn, that you learned from COVID-19, from Mpox, that we can apply towards bringing an end to HIV, to TB, hepatitis, and whatever else you're working on. Thank you.
That was really great, and actually I did want to start a discussion or any questions to Dr. Kim. Please.
I just have a quick question in terms of getting ahead of the pandemic. Does wastewater surveillance give you any sort of meaningful edge, or is that lead time too marginal to really make a difference in the implementation of the trial?
Yeah, we have found that I personally believe it's too marginal. Yeah, because basically it's just a real-time detection. It's not any kind of predictive focus. So, and actually also we're finding that it's not always predictive. So, I think again, those are the early indicators. It's better than not having it. But I think if we really want to be prepared, well, what I feel is that we already need to have, by the time we find it in the wastewater, that site already needs to be activated and ready to go. Yes.
So Peter, we're so lucky to have you, and I don't think you can work more than 16 hours a day. So one of the questions I have is, there are some pieces of the clinical research enterprise, and one of them are these CROs, okay? And so why can somehow, it's a contract, why can't that be reduced from 4 months to 1 month? Can't you incentivize them to just rapidly reduce that process? And I know it's like, no, the FDA says we can do this or that, but really somebody's got to take responsibility for that, because that's out of your control in a lot of hands. And it really is a big burden on people doing research.
Yeah, so can I just clarify, when you say 4 months, what aspect are you
Just
referring to?
site, getting sites up and going, all the paperwork, all the meetings, all the stuff that has to be done. And a lot of wasted time stuff, honestly,
Right.
in terms of our goal to find out if something works, keep our patients safe,
Yeah.
and getting data that the FDA will accept
Yeah.
for approval.
Yeah, absolutely. And actually, I would have to say, the FDA has nothing to do with this, because the FDA asked me the same thing. Why can't you get CRO to activate these sites? What we found is that in certain instances we are. So one of the, I think, the strengths of the DAYS clinical trial system, the ACTG, HBTN, IMPACT, HBTN, is that we actually maintain a cadre of sites, so they're ready to go. And actually, we are currently working with clinical project managers from industry, and they have expressly told me that they have seen the strength of this, because they're seeing that we're able to activate sites much more quickly than actually you see in industry. Having said that, there's only we did push, trust me, we pushed these CROs as hard as we possibly can, and Annie is a testament, to what we're doing even now. But what we're seeing is that, number one, there's only so much work that can be done on the CRO side, because half that work depends on the site actually responding to the needs. And again, many sites don't respond on a timely basis. We just had a call this morning about the MPOX trial, where certain sites have told us early on they want to participate, but after months, there's no response from them. All they need to do is fill out some simple forms for them to participate in the trial. So number one is really engaging with the sites and getting them to do their part. Number two, what we found is that CROs actually have all of their, there's like a million little things that they have to do, and even with all the resources that we could muster, there was only so much that we could push them. So I agree with you, and I'm constantly thinking about actually how do we do this better, and I do think it's something that we have to think about for the future pandemic, because obviously it's such a critical time gap, right? 1 month versus 4 months, and 4 months is just the average. It's not even like the full duration. There are many sites that take 6 months or longer to get activated in the midst of a pandemic. So yes, we need to think outside the box, I think, as a research community. How do we do this? Because right now, the onus and burden of getting a site activated is it's too much. And we need to think, how do we do this better? I agree. Okay, Monica, can I just?
You just please.
Oh, okay, well, Annie had her.
Oh, yes.
We'll go this way.
Thanks. So I couldn't agree more. And I think one thing that I really saw was that having all the clinical trial sites ready to go and then get leveraged. I mean, we all saw CoVPN that didn't exist, and all of those vaccine trials were stood up and completed very quickly through CoVPN, and it involved new sites. Same thing with the ACT studies, and that NIH was able to be flexible, but you have to have those sites that exist. You can't be like, oh, I got to hire people. That's not going to happen. But I think on top of Diane's question, I was heartened by the COVID pandemic to see some things that we'd taken years to bang away at finally went away, right? Like our institution said, fine, you can use DocuSign to consent people. We've been arguing with them for forever. We can enroll people remotely. You can do remote monitoring, like a lot of things that we'd all been pushing for. But some things I'm worried about backsliding now, or that we can't keep doing that. The FDA is part of the problem. I remember at one point during the pandemic, they came to me and many other sites and said, well, we really want community hospitals to be involved in this research. I said, you're the problem. Community hospitals don't want to be part of this research because they don't want to fill out a 1572. They don't want to do all this stuff. I don't blame them. Are you going to change what you're doing? And they never called me again, so I don't know. But I do think they have to see themselves, when even we start with that forms are called 1572 and did you do your blah, blah, blah. It's like, that's going to be, I love what you brought up about, yeah, most of our sites are urban, academic, and that's a good thing. But we gotta have like rural representation. We really miss the boat there. And so I think unless we change our systems, we're gonna get stuck with the same groups enrolling, and that misses the community. And then the group I'd love for you to address, in addition to, more rural, more community-based, is international, because that's a thing where I see that going only in the wrong direction. And there was like this moment where maybe you could get local ethics committees to say, oh, this is COVID, but now that's gone away. They're like, well, you're to the back of the line. It's appalling. And speaking, they really don't have anything available. There's no treatments in many countries. This really is still the only way for people to get access to some therapy, and their countries just, I feel like we're losing traction there. So I love your perspective on how we can speed up ex-US and the FDA and the community.
Yeah, well, number one, I agree with you, and to just add a punctuation mark there, an exclamation point to what you just said. I mean, there were some countries that, again, early on in the pandemic, there were no treatments available in that country, and still they could not, find it amongst themselves to approve our trial in a timely basis so that we can open there. I mean, I think these issues are, when I talk to my scientific, collaborators and even these governments, I mean, they understand the issue. I think these are more political than scientific issues. So I bang my head. I'm constantly thinking about how do we make this better, but I don't know that there is actually a scientific solution to that. I think it has to be a political one, except for there is one actually idea that I'm a strong believer of, and that's the idea of federated data science. And I'm glad bioinformatics is a key new core here, because I do think that we're living in a world where increasingly countries want control over the data emanating from their countries, whether it's data or laboratory, results. And I think we need to develop the systems where we can enable data and laboratory data to be handled in country, but we, at the end of the day, for accumulated analyses, have that happen in a central location. And I know that this is an idea, it's not my idea, it's been circulating for quite a long time. But if you think about actually how long it's been circulating and how much of it actually has been implemented, it has not been implemented at all in HIV, TB, or hepatitis circles, as far as I know, and even all of biomedical science. I think it's something that's being done more like Google and other companies for commercial purposes. But I do think it's something that we really should undertake, because I think it'll go a long ways to showing countries and people that may still distrust us that we're not here to take or steal anything from you. What we're trying to do is help. We're trying to advance science, and if we can do it in a way that doesn't impede your oversight and your ownership of the data, we will. So, okay, Monica, and then up there, and then we'll go here. Yeah.
Well, I mean, I actually wanted to bring it up to a different level, which you touched on, which was the bipartisan support. And I think that Dr. Fauci's greatest strength in the HIV epidemic was being able to bring bipartisan support to issues, and from the NIAID level. And that didn't actually happen during COVID. And when, for example, PEPFAR reauthorization is up, and you're watching all these pieces in both the literature and newspapers, they're all really derogatory towards Republicans. They literally, the New England Journal had a piece on Sunday that said they're trying to ruin everything. And that's actually not a good way to win friends and influence people is to say that someone's ridiculous. And we have a piece coming out that's trying not to do that with PEPFAR. But do you, when I was with you the other day, there's such a clear bipartisan difference in how NIAID may be supported. Can you speak to not being so derogatory and trying to bring that bipartisan support back for HIV? Because it seemed like after an initial really hard start, there was, PEPFAR was formed by Bush. There was a bipartisan aspect of this, and we seem to be getting away from it on this side as well by saying derogatory comments towards people.
Yeah, no, I would wholeheartedly agree with you. I mean, I think science historically has been not a political issue, and we have the NIH has enjoyed support from all parties. And I hope that we can get back to that. Because again, I mean, science is science, and it shouldn't matter what your political views are in your support of science. So I wholeheartedly agree with you that it doesn't help attack people based on their political beliefs, and we should not be doing that.
Oh, great. Thank you so much for this. This was so fascinating. Sophia, again, have to admit, as a qualitative researcher, I thought that this was potentially going to have a personal angle just with the title, which I loved. But I wanted to go back to the slide where you talked about even at the sites that had established and enrolled, had the potential to enroll participants, there was still a lot of them who were not. And I'm wondering, you had mentioned there were a lot of things that went into that, but what do you think, was there some way to, it sounds like it's even study staff, was there a lot of, what's going on there? I'm just curious. This is the only way to get treatment. Cases are happening, deaths are happening, people aren't enrolling even at established sites.
Yeah, so I have no data to support this, so these are just my hypotheses, but there were multiple things happening at this time and even now. So number one, as I mentioned, there were, even though we were the main study for this country, there were, over 1,000 trials going on, small single site studies that were happening. And I suspect, number one, that many sites actually, were trying to enroll into these small studies together with our study. And again, if you're an investigator and you have your own study, or you have your colleague study to enroll into versus this kind of NIH study, but you don't know the people who are running it, you might be pushed to enroll one. Second thing is that I do know that there was a healthcare resource shortage, right? And there was just not enough nurses and doctors to go around at that time. So I do think that in some instances, sites had to make the hard choice. Are we going to put our resources to taking care of patients in the ICU and on the wards, or enrolling into the study? And there was just not enough resources to go around. Third, I do think that we also know there was fragmentation even amongst the research community about what the best treatment. So there were many people that actually felt new drugs, that's crazy. Let's repurpose drugs that we have ongoing. So they just didn't agree with the tenor or the goal objectives of our trial and felt that their resources were better utilized towards other places. Those are the top 3 things I can come up with, but this is one of the things I would love to hear from you about, because I'm not at the site level, but help me understand what were some of the things that were going through your minds, and when you had to activate and enroll into these studies, because I think that's critical for us to understand. Okay.
Well, I was actually going to ask the same question. It's really interesting from a social science point of view. So with the enrollment, was it the people in charge of enrollment who were refusing to enroll their patients, or was it patients themselves who were refusing when they were asked if they would enroll?
Yeah, I really have no idea. I mean, I think I don't want to take this too far and say that those sites were turning away patients when patients were clamoring at their door. I think that would be one step too far for me to say that. I actually don't know what was happening at that site level, and I suspect that there were a number of issues that impacted their inability to enroll.
There's a comment online.
Payam, do you want to give your?
From Payam this, you can.
Oh, sure. Yeah. Hi, Peter. Thanks very much for that provoking talk. I know this is something you're quite passionate about. First, I'll start with the obvious, which is, as everybody here knows, we do have a major pandemic ongoing, tuberculosis, killing more than 4,000 to 5,000 people a day. And we're pretty damn slow about our protocol launches for trials for that condition. And I know that you share the frustration there. I guess my broader point is whether it's a pandemic that we just dealt with in COVID or an ongoing pandemic, or frankly, the next one that we have no idea what it will be. I am feeling a bit cynical. I'm not sure we've learned our lessons. I will say that there's probably some things we can do to harmonize the lessons, and I'm wondering whether you have any thoughts about that. I can tell you that at UCSF, like Annie mentioned, we triaged COVID trials to the top of the list, really moved all the administrative aspects aside as much as we could, IRB, new tools. And in conjunction with that, our faculty submitted nearly 600 proposals for COVID-19, $700 million of funding was requested. I think we got about 150 million of that by around November of 2021. And we had 30 active interventional COVID trials. And you're right, there was a major problem with workforce, with the infrastructure really creaking and ability to handle both the clinical burden and the research needs. And you mentioned informatics and data systems and surveillance systems, but I think the next pandemic that hits, I'm not sure we're not going to find ourselves in the same place. You know, lots of proposals being submitted, rapidly funded, 20 to 30 trials activated, and a workforce that's really straining. So what can we do better? I mean, I think everything you said I agree with, but I'm not sure we've learned what to do and where to take this discussion further.
Yeah, I mean, that's a good question, Payam. If I understand, I will admit I had a little bit of difficulty hearing everything, but I think you're basically saying how do we approach this heterogeneity problem in the U.S.? I mean, I think the strength of the U.S. scientific community is that we're heterogeneous, right? And that we have freedom to approach our scientific interests. But that is, in a pandemic, that was also our weakness. I can tell you that the NIH did not fund any additional clinical trials, any of these small clinical trials. So the funding for that did not come from the NIH. I can also tell you that I was frequently, constantly on calls with other major funders like the Gates Foundation to coordinate on our trial. So I don't know where the funding for these other smaller trials came from, but I hope that actually, those funders hear the message about this. The second thing, I think we have to go to the base of investigators, right? And we as a research community need to agree that in the setting of a pandemic, in a setting where what we need is a centralized effort and not a diversified one, we just have to agree that we're going to work together. And obviously, much easier said than done, right? Because how do we, do that? I don't think, at least I don't have the stomach, I don't know that anyone else does, to put limitations on people to do that. I hope that this is something that we as a research community can come together and work together on and show ourselves, and do it ourselves rather than it being forced down our throats. Because again, I think a strength is that heterogeneity, and I would hate to lose that strength just because of this potential harm in the setting of a pandemic. Those are my thoughts with that. But I'd be curious again to hear your thoughts from the research, from your perspective of UCSF. What do you think would work? And how should the NIH next time, hopefully there is no next time, but if there is, how should we approach universities and academic groups? I can tell you that I was sending out letters from Francis Collins. I was sending out letters from Dr. Fauci. I was sending out letters signed on my behalf, basically imploring sites to participate in our study, to enroll in our study, and doing all these things. I can tell you that had very little effect. To my surprise, that had very little effect. Basically, universities and academic groups did not want to be told what to do. It's their right, but again, that ultimately, I think, detracted from our national effort.
We should end, I think, unless are there more on the chat?
There's no more.
Okay, I thank you so much, Dr.